Welcome to another video on biology revision. In this one we're revising infection and response, the third topic in Biology Paper 1. It's all adapted to the 2022 exam advance information. Please subscribe if you're finding these revision videos helpful and share them so that other people have a way of revising. There's barely more than a week left until the exam, so any revision you can do is useful. And now we're going to go and look at the advanced information. So, Combined Science Foundation, your focus is communicable diseases, so the first two slides, 4.3.1, nothing from this video has been taken out, apart from obviously the triple science only bits. There's no required practicals covered in this video either. So Combined Science Higher, this isn't a major focus of your exam, and viral, fungal and protist diseases as well as human defense systems have been taken out, but the rest of it could be examined, you don't know, but these four you can skip because they're definitely not coming up. If you're taking biology as a single science, or triple science as I call it, again, communicable diseases, 4.3.1, the first two slides, is your major focus, and Protist diseases has been taken out for you. And finally, triple science higher, communicable diseases is your major focus, as well as monoclonal antibodies. They will be examined in your exam. And for you, antibiotics and painkillers and the discovery and development of drugs have been taken out. All the bits that have been taken out are highlighted. We're now going to get into the video. So remember, for this first slide, all the stuff in purple will not be for Combined Science Higher, and all the stuff in pink will not be coming up in the Triple Science Foundation exam or the Combined Science Higher exam. So we're going to start this topic with an in-depth look at diseases. So here's our first 7 questions on the spread of disease, if you want to pause and have a go. Pathogens are simply microorganisms that can cause disease to humans or animals. Pathogens can be spread in 3 different ways. First of all, through the air, like COVID is. Obviously, if someone sneezes, you inhale the droplets that they sneeze out, then the pathogen is spread to you. Water as well. I'm sure you've all heard of waterborne diseases like cholera. And also through direct contact, so like touching the same things as an infected person. So here are the 4 main types of pathogens that you need to know about: bacteria, viruses, fungi, and protists. Bacteria, they're prokaryotes, as we've covered, and they enter our body and reproduce very rapidly due to our body's optimum conditions for binary fission, and they make us ill by producing toxins. Viruses, they aren't actually cells, so it is a debate about whether they're living or not, but basically they live inside our cells, they use our cells to clone themselves, and then they cause our cells to burst, and all those viruses, those viral particles, leave the cell and find new cells to make lots of replicas of themselves, and it's this cell damage that makes us ill from viruses. Fungi can be single-celled, they can be hyphae, which are like lots of little string-like structures. Basically, they land on our skin, our feet for example, and they penetrate into our skin using their hyphae, so they kind of grow into it, and this makes us ill. And protists, or protists, they're eukaryotic cells, and they are parasites, so they will live inside our body and cause us damage and make us ill by using our body's resources. Preventing the spread of disease is something you should be very familiar with over the past few years. There's 4 ways you should be able to name. So, for example, isolating infected individuals, like we've been doing for, it feels like ages. Being hygienic, washing your hands using hand sanitizer before preparing food, for example. Vaccination, which we'll look at later. And the final one is one you may not be familiar with: destroying vectors. Vectors are basically organisms or things that spread disease. So I'm going to give you an example. Malaria is a protist spread by mosquitoes. Mosquitoes are the vectors because they spread malaria by biting us. They bite us, they put the malaria inside us and it causes us harm, but the malaria doesn't actually do any harm to the mosquitoes themselves. There's 3 viruses that you need to know about, so pause the video and have a go and see how many you can remember. Measles is spread through the air, so it's spread by droplets. When people cough or sneeze, those droplets, people can inhale them, breathe them in, then they have measles. Uh, Measles is typically characterized by a fever and a red rash, so fever being a high temperature, coughing, sneezing. And measles can become pretty serious. It can be fatal in some people. It can also lead to pneumonia, which is a lung disease, or blindness or seizures. Yeah, it's very serious. We treat measles by vaccinating children, so it's not really much of a problem anymore. HIV is spread through direct contact, mainly sexual contact. It is a STD. The symptoms of HIV, after you've got it, you may have some flu-like symptoms for anywhere from a few days to a few weeks. So some coughing, sneezing, maybe a temperature. But then, interestingly, for a few years, you don't get any symptoms at all because the HIV is in the body. You doesn't affect you directly, but it's targeting those immune cells, so it targets the immune system, and this means eventually it can lead to something called AIDS, where the immune system is so badly damaged because all your immune cells have just been infected and burst by this HIV virus that you can't fight against any other infectious diseases, and that's AIDS. So we treat HIV. The best time to treat it is ironically when you don't have any symptoms, and in that time we can use drugs that prevent the virus from replicating and destroying your immune cells. Finally, a plant virus, tobacco mosaic virus. It's spread through, again, direct contact through infected gardener's tools or human hands that have touched a plant that's been infected by TMV. And the name explains the symptoms. The main symptom is this mosaic pattern on the leaves of the plant. This mosaic pattern causes leaf discoloration, which means that the leaves aren't green anymore, so they can't carry out photosynthesis. So tobacco mosaic virus kind of stops growth as well in the plant. So here are 2 bacterial diseases you need to know about. Pause and have a go at these 6 questions. Salmonella is a type of food poisoning, so it's spread through food. It originates from chickens, so chickens originally have it, but if we eat, say, chicken eggs or chicken itself from an infected chicken, then we get it. So that's how it's spread. And its symptoms, usual symptoms of food poisoning: diarrhea, vomiting, stomach cramps, the usual disgusting stuff. There's fever as well sometimes. And we prevent the spread of salmonella through vaccinating chickens, which means that if the chickens don't have it, then we can't get it from eating the chickens. Gonorrhea is the really gross one. So gonorrhea is an STD, sexually transmitted disease. You get it from unprotected sex. Its symptoms: pain when you urinate, as well as there's this thick yellow liquid discharge that really disgusting that comes out of the penis or vagina. Yeah, you're not going to forget that. And we prevent its spread. We can use antibiotics, but bacteria are becoming resistant to antibiotics. So condoms are a great way to reduce its spread, as they prevent the exchange of fluids during sex. If you want to pause the video now and have a go at these questions on fungal diseases. So the one fungus you need to know about is rose black spot, which affects rose bushes. So rose black spot is spread through wind or water. Its symptoms are, well, quite obviously, it causes a black spot. So if you remember, that fungi lands on the plant's leaf, it puts down its hyphae into the plant, and it appears as a black spot. And its effects: it can cause the leaf to turn yellow and drop off, which decreases the amount of photosynthesis that can happen in the rose plant, and this can affect its growth. And gardeners can treat rose black spot. We can use fungicides, so just liquids that kill fungi, or we can destroy the affected leaves, so pick them off and destroy them so that the fungus doesn't spread to any other rose plants. So finally, the Protist disease you need to know about: malaria. Pause the video and have a go at these 3. So malaria is spread through mosquitoes, as I gave you an example of earlier. The mosquito is a vector, so what that means is the malaria, the protist, that eukaryotic cell, it's living inside the mosquito, but it's not causing it any damage. But when the mosquito bites us, it transfers malaria to us The malaria then lives inside us and causes us damage, and the symptoms are predominantly a fever, so a high temperature, headaches, being sick. It can be fatal. It's a serious disease, so it needs to be treated quickly. And we can reduce its spread by preventing mosquitoes from breeding and killing them using insecticides. And we can also encourage people to wear, and ourselves, we can wear long sleeve t-shirts and long sleeve tops and trousers to prevent mosquitoes from biting us, and we can put mosquito nets up in our homes. But remember, it's not in the UK, it's mainly in really warm countries and continents like Africa. Here's another quick reminder of which bits have been taken out of which people's exams. So Combined Science Higher should avoid purple, Triple Science Higher should avoid the orange. Now we're going to look at how the body defends against pathogens, so pause the video and have a go at these four. So the body's non-specific defense systems, non-specific simply means they defend against any pathogen, no matter what. So the first one is a skin. Our skin just acts as a barrier, prevents any pathogens from getting in, and it also secretes antimicrobial substances to kill pathogens. The second one is our nose. So in our nose we have hairs, we also have snot. These trap particles that could contain pathogens. The third non-specific defense system is the trachea and bronchi in our lungs, so our windpipe that goes down to our lungs. These secrete mucus, your phlegm that you phlegm up. These trap pathogens, but in both the snot and the mucus, we then get it back out the body, so the pathogens come back out the body. And our fourth non-specific defense system is the stomach, because it produces hydrochloric acid. If you remember from the last video, that is very acidic, so it kills any pathogens that fall into the stomach. So that's question 2 answered as well. The main part of the body's immune system, the white blood cells, these are essentially, they do everything to stop pathogens, attack pathogens, destroy pathogens. The first way they fight pathogens is by consuming them. So if they come across a pathogen in the body, they will engulf it and digest it, and this process is called phagocytosis. A nice complex name for you to learn there. The second way is they produce antibodies. So these antibodies, they lock onto antigens that basically just proteins on the surface of these pathogens. They attach to these antigens, they then alert the white blood cells to come over and engulf the pathogen by phagocytosis. It's also worth remembering that these antibodies are specific, so each antibody will only attach to one specific antigen, and when your body has antibodies that can attach to the specific antigens, then you're immune to the pathogen that contains those antigens. And finally, the third way is the body produces antitoxins, and antitoxins basically combat and destroy the toxins produced by any invading bacteria. Obviously, if you remember, bacteria produce toxins which make us ill. Now you can pause the video and have a go at these questions on vaccinations. So when you're injected with a vaccine, you're essentially injected with the pathogen that the vaccine protects against. That pathogen is going to be inactive or dead, it's not going to be live, but the key thing is that the antigens of that pathogen in the vaccine, because your white blood cells will sense those foreign antigens and they'll come over and they'll produce antibodies to attach to those antigens. But the key thing is that when your white blood cells first see new foreign antigens, it takes them a while to produce the right antibodies to attach to those specific antigens, because antibodies are specific. So that's why you get pretty ill, because when your body gets infected with a new pathogen, it spreads around the body, causes damage, but your antibodies are still your white blood cells are still trying to figure out how to produce the antibodies specific to the antigen on that pathogen. So when you're vaccinated, your white blood cells figure out how to produce the antibodies specific to that antigen, so that when you're infected with the actual pathogen, your white blood cells remember how to produce the antibody specific to the antigen on the pathogen. So they produce those antibodies, they attach to the antigen, mark out that pathogen, so your white blood cells come and engulf them all. And usually this is before you even get any symptoms of the disease and even know you've had it. So vaccines can protect a whole population from a disease, because when everyone's vaccinated, there's less or no time when your body is infectious enough that you can, for example, sneeze and pass on those droplets of the pathogen to other people. And even if someone else does catch it, once again, they're not going to become particularly ill from it, if at all. And once again, there's less or no time where they can pass it on to other people. So there are less people getting it. And even if you're unvaccinated, you're much less likely to get it, because there's loads and loads of people that have a really low chance of passing it on to you. The downside to vaccination is that some people can get bad reactions to it. So some people can get swelling, it can make people feel pretty ill or fevery for a day, but these are relatively rare. Pause the video and have a go at these 6 questions on antibiotics and painkillers. So antibiotics are simply medicines that help cure bacterial disease by killing infective bacteria inside the body, and the key is the pathogens they affect is bacteria. Antibiotics do not affect viruses. However, some bacteria can become resistant to antibiotics by mutating, and we'll cover this more in paper 2, but when they mutate, then they could become resistant to an antibiotic, so they don't get killed by it, which means that only the non-resistant strains of bacteria will be killed, and the resistant strains will survive and continue to damage the body. And there's a number of things we can do to reduce the resistant bacteria, although to actually kill them inside the body is much more of a challenge, and this is why antibiotic-resistant bacteria are a serious concern. We can reduce new strains of resistant bacteria by not overusing antibiotics, because the less bacteria are exposed to antibiotics, the less chance they have to mutate to become resistant to antibiotics. It's also important to finish the whole course of antibiotics and not overtake them or undertake them, take them exactly as the doctor has prescribed. So the difference between antibiotics and painkillers and other medicines: painkillers relieve pain, obviously other medicines relieve other symptoms, whereas antibiotics, they actually kill the pathogen. Other medicines don't, they just reduce the symptoms from the pathogen while your body fights the pathogen. And we don't have virus-killing drugs because it's very difficult to develop a drug which can go into cells and kill off those viral particles without killing the cells that all the viruses are inside, because if you remember, the viruses go inside and clone themselves using your cells. So here are 11 questions on the development of drugs, so pause and have a go at these. Digitalis was developed from a chemical found in foxgloves. Aspirin was developed from a chemical in willow bark, and penicillin was developed from a mold that was found to kill bacteria. And the function of digitalis is to treat heart conditions. Aspirin can be used as a painkiller, and penicillin is an antibiotic. It was in fact the first antibiotic that was used. Today, we develop drugs from synthesized chemicals in the lab, although we may still start with a chemical from a plant, but we usually edit it, synthesize it, whatever. You don't need to know any details about that. We need to test drugs to make sure that they're safe for people to have and they're also effective. And following on from that, we test drugs for toxicity, if they're toxic to the body, efficacy, how effective they are, and the optimum dose, so how much you need to give someone. In preclinical trials, we start off by testing on human cells and tissues to make sure it doesn't destroy cells and tissues, because that would obviously be bad. Obviously, there's no point testing on human cells and tissues if we're testing a drug for blood pressure or something. So we move on to the live animals test in that's the second part to the preclinical trials. Some people think that it's unethical, but it is what it is. This is to mainly test for toxicity. Clinical trials start by testing the drug on healthy volunteers, so you'll inject a very small amount and then you'll gradually increase it to make sure it's not toxic. And then once we've made sure it's not toxic, to humans, we go on and test it on patients who are ill from the pathogen that the drug will kill or prevent against, because only antibiotics kill bacteria. The others don't, remember, that's very important. And we test this drug on patients to make sure it's actually effective and it does its job, because if it doesn't, there's no point in it having it in the first place. And when we know it's safe and effective, we can do a final clinical trial to find the optimum dose of the drug. These days, we use double-blind trials in clinical trials to test how effective the drug is. This means splitting the patients into two groups. One of the groups is given the actual drug, one of them is given a placebo. And the placebo is a substance that's like the drug, but it doesn't have any effect at all on the body. So this stops the placebo effect, which is where the patient expects the drug to work and therefore it works, even though the actual drug itself hasn't done anything. It's quite a weird psychological effect, but there we go. And in double-blind trials, neither the patient nor the doctor knows which group has been given the placebo and which group has been given the drug to be tested. And this prevents the doctor from being subconsciously influenced by their own knowledge or opinion on the drug. And the final stage of drug testing is peer review, so other scientists will check over the work and the trial and make sure it's all okay, prevent false claims, make sure it's all official and everything. And that's how drugs are developed. So these questions on monoclonal antibodies, this topic has only ever been in the Triple Science Higher exam. So if you're Combined Science or Triple Science Foundation, you don't need to worry about this at all. So pause the video and have a go at these 10. So monoclonal antibodies are made from a single clone of white blood cells. And if you remember, antibodies and monoclonal antibodies, they're specific, so they only target a specific protein antigen. But the key is that they've been made from lots of clones of a single white blood cell. The white blood cells that produce monoclonal antibodies are known as B lymphocytes. So producing monoclonal antibodies in the lab starts with a mouse. You're going to inject that mouse with the antigen that you want your monoclonal antibodies to target, and that mouse, the mouse is white blood cells, the mouse is B lymphocytes, they're going to produce antibodies to bind to that specific foreign antigen, and then you're going to take out the B lymphocytes that now know how to produce the antibody for that antigen. And to answer question 4, we need a tumour cell. What we do is we take those B lymphocytes, we fuse them with a tumour cell to make something called a hybridoma. And why we need a tumour cell is because the white blood cells, they divide very slowly on their own. But if we take a tumour cells are very fast dividing. So we fuse them and we have a cell that can both produce the antibodies that we want, and it can also divide very rapidly. So once we've got our hybridoma, which is the name of the tumour cell and the B lymphocyte fusion cell, we clone it a lot, and therefore we get lots and lots of cells that can produce the same type of antibody, these monoclonal antibodies, and we can collect these antibodies, purify them, and they're ready for use. So one of the main uses of monoclonal antibodies is in pregnancy tests, because we can produce monoclonal antibodies that bind to a hormone called HCG, and HCG is found in the urine of women only if they're pregnant. So we can test for this hormone, and we test for it. What we do is we take these monoclonal antibodies, we attach them to blue beads, and put them on like the pregnancy testing strip, whatever, and then the woman has to urinate on the strip, and what happens is the urine and the beads with the antibodies attached will move up the strip, and a bit of the way up the strip there's going to be a line of antibodies stuck down. Now, if HCG is pregnant, when the woman urinates on the strip, the HCG hormone will become attached to the antibody which is attached to the blue bead, and when they move up, that hormone attached to the antibody attached to the bead will become attached to the antibodies stuck down. So you'll have an antibody stuck down which is attached to the HCG hormone, which is attached to another antibody which has a blue bead on top, and all those blue beads will come to rest as they're attached to the strip by the stuck down antibodies, and a blue line will form, and that shows a positive result. If there's no HCG, then there's no hormones to bind onto the antibodies and glue the blue beads to the test strip where the antibodies are stuck down, so the blue beads are just going to move up to the top and no line will appear. Monoclonal antibodies can also be used to treat cancer, so they're actually very important and used quite a lot. So what we can do is, again, we can make monoclonal antibodies that attach to the antigens on the surface of the tumour cells, the cancer cells, and they are different from the antigens of normal cells. They're called tumour markers, the antigens on the surface of tumour cells. So what we do is we make these monoclonal antibodies that attach to the tumour markers, and to these monoclonal antibodies we can attach an anti-cancer drug that can kill the tumour cells or stop them from dividing, and then we put these antibodies into the body and they'll go to the cancer cells, they'll attach to the tumour markers, the antigens on the outside of the tumour cancer cells. The anti-cancer drug will work, it'll kill the cancer cells but leave the normal cells unharmed. There are other uses of monoclonal antibodies as well. We can use them to test for substances in the blood. So again, we'll make monoclonal antibodies that bind to the antigen for that specific substance that we want to test for. It may well be a hormone or a pathogen, for example, or maybe another chemical. And instead we put those antibodies in, and we are then able to see how many of them bind to the substances, and we can find out the level of that particular hormone, chemical, pathogen, or to whether the pathogen's present in the blood. Also, we can use them to test for specific molecules in a cell. So what we'll do is we can bind them to a fluorescent dye, these monoclonal antibodies that we've made to test for the substance that we want to test for. We put a fluorescent dye on them. We can then insert them into the cell, and they will bind to the substance we're testing for in the cell if the substance is present, and that fluorescent dye will show up and show that they've all binded to that substance, and therefore that substance is present. The big advantage of using monoclonal antibodies to treat diseases such as cancer is that they specifically target the tumour cells. So they will bind to the tumour cells, but they will not bind to the normal cells. That's very key. So they won't damage the normal cells, they will only damage the tumour cells, and that means that they have less side effects than other cancer treatments, like chemotherapy, that has more side effects than using monoclonal antibodies to treat cancer. There are ethical issues associated with them. If we think right back to the mouse that all these monoclonal antibodies come from, first of all, we have to inject the mouse with either a harmful pathogen or a drug containing foreign antigens, which will it will damage the mouse, harm the mouse, and then to extract the B lymphocytes, the white blood cells that know how to produce the antibodies we're looking for, we have to remove the spleen of the mouse, and this kills the mouse. So many people are against animal testing and using animals, and each clone of monoclonal antibodies that we want, we have to sacrifice a mouse for it, and that's why there's ethical issues. They also aren't as widely used as scientists first hoped, and this is because they have much more severe side effects than scientists expected, because we thought that, because they're only going to bind to specific cells, because they're only going to bind to the tumour cells, they won't have any side effects because they won't affect normal cells. While they do have less side effects than things like chemotherapy, radiotherapy, they still do have quite bad side effects. like vomiting, low blood pressure, for example. So that's why they're not as widely used in treatments. So our final section is plant diseases, and once again this topic is only in the triple science specification, so if you're combined science you don't need to worry about this. If you want to pause the video and have a go at these 13 questions, these first two are higher only. So, there are many ways we can detect a plant has a disease. The main way is by looking at the leaves. There could be spots on the leaf. The leaves could be rotting. There could be like fungal growths on the leaf. The leaves could be malformed. They could be discoloured. For example, tobacco mosaic virus has that mosaic pattern on the leaves, or leaves often can turn yellow and brown and drop off. For the plant as a whole, the stem could become malformed. There could be growths on the stem. It could be stunted growth. The plant could not be growing at all. Or there could be pests as well. And by using the signs of plant disease, this detection, we can identify the specific disease a plant has, or if we've just been watering it too much, and any plant owners will know my pain. So the main way of identifying a plant disease is using a gardening book, manual, or going onto a gardening website, looking up the symptoms of a disease and seeing if it matches the symptoms your plant's displaying. But if you're a member of the Royal Horticultural Society and you're looking after prize plants and they all start having a disease suddenly, you might take the infected plants to a laboratory. That's the second method. And in the lab, they will identify the pathogen using monoclonal antibodies, actually, funnily enough. So again, you can see another use of monoclonal antibodies identifying pathogens in plants this time. And the third way is you can purchase monoclonal antibody test kits to use at home, so you don't actually have to pay loads to send them off to a lab and waste scientists' time identifying which kind of disease a plant has. You can just buy these test kits with the monoclonal antibodies and identify it yourself. So those are the three ways of identification. So if we think back to tobacco mosaic virus, we covered this at the beginning. If you remember, it causes that mosaic pattern on the leaves, discolours the leaves so that they aren't green, they can't absorb sunlight, so less photosynthesis happens, and therefore less growth happens. Rose black spot is a fungus that affects rose plants and rose bushes, and if you remember, it causes black spots to appear on the leaves, and then the leaves turn yellow, they drop off, and this means that less photosynthesis can happen because obviously there's less leaves, the leaves are yellow, and the fungal growths, the spots, that no photosynthesis can happen there. And if you remember, we treat rose black spot by taking the leaves off and using insecticides. And finally, aphids. Insects and pests such as aphids, they can infest plants. What aphids do is they can infest plants in large numbers. They can drink the sap of the plant. They get lots of nutrients from that. So they'll drink the sap of the plant. They'll take away all its nutrients, and this stops it growing because obviously it's losing loads of nutrients. And this eventually kills the plant. And some farmers some farmers and gardeners can use ladybirds, which eat aphids, or insecticides work as well, but we're trying to be more natural and environmentally friendly, so ladybirds are often better. Nitrate ions are needed to make the amino acids for protein. If you remember, amino acids are made up of a nitrogen compound. So plants suck up nitrate ions from the soil to make their proteins, and without their nitrates, a nitrate deficiency can lead to stunted growth because they don't have enough proteins, and proteins are used for growth. Plants suck up magnesium ions through their root hair cells, and these are used for making chlorophyll. And magnesium deficiency, a magnesium ion deficiency in a plant can cause chlorosis, which essentially means the plant can't make any chlorophyll. And if you remember chlorophyll, that green pigment that absorbs the light for photosynthesis, this means the leaves turn yellow, photosynthesis happens. Photosynthesis can't happen, and therefore growth is stunted as well. So now we're on to plant defenses. Physical defenses are an adaptation that the plant has actually grown. It's something that it actually has, something physical that it actually has. A chemical defense could be like a poison or a chemical to deter predators or pathogens from entering it. And a mechanical adaptation is like a mechanism, something that the plant does. So ferns curling in on themselves is a mechanical adaptation. So physical defenses, there are three of them. The first one you can remember is that all plant cells have that cell wall made out of cellulose, and this is just a physical barrier against pathogens. It stops pathogens like viruses, for example, from being able to enter cells and enter tissues and organs, enter leaves, and cause loads of damage. There's also that waxy cuticle, if you remember, over the surface of leaves. That's similar to our skin. It stops pathogens from being able to enter the leaf. And there's also layers of dead cells around lots of plant stems. For example, trees have bark. That's just a layer of dead cells to keep pathogens out. It just acts as a physical barrier which protects against them. Remember, that's similar to skin, once again. Chemical defenses, there are two. Some plants produce antibacterial chemicals, which simply kill bacteria or pathogens. And plants can also produce poisons that can deter herbivores from eating the plant, because obviously no plants want to get eaten. So, for example, foxgloves have a poison. Don't eat foxgloves. Don't want to get poisoned. And finally, the mechanical defenses. There's three mechanical defenses you need to know about. I'll give you an example. The thorns on the rosebush, some hairs, they can prevent animals from touching plants and getting close and eating plants. For the ferns, they droop and curl when something touches them, and this means they prevent themselves from being eaten. It knocks insects off. They move away from maybe the herbivore that's trying to eat them. They just move away. And some organisms perform mimicry. And for example, there's one plant that has bright yellow spots on its leaves to look like a certain type of caterpillar egg, and this stops the butterflies from laying their eggs, which hatch into caterpillars which eat the leaves of the plant, because it looks like there's already loads of caterpillars that are going to come out and grow, but in fact there aren't, so the leaves stay safe from caterpillars. And that's what mimicry is. You mimic other organisms. So there we are at the end of the infection and response topic. It wasn't quite as big as the other ones, which was quite nice, especially if you're doing combined science. There wasn't really much to remember there. More triple science only content. But yeah, if you went along and did the questions, any questions that you got wrong, come back tomorrow and have another go, because that's how you're going to remember. By redoing the questions you got wrong, then you're going to get them right. The knowledge will remain in your head for the exam, which I believe is in roughly 8 or 9 days from now. So revision is very important. Come back to these questions before the exam again, just to make sure that you know them all. Remember, only do the ones for your exam. Don't waste your time on ones that aren't going to come up. And also practice some exam questions, I would recommend, just so you know you're going to secure those high grades, but you know you've got yourself fully covered. Here are the slides of questions. There's the communicable diseases slide with an introduction to the different diseases. There's the fighting diseases, and then there's the monoclonal antibodies and the plant diseases for triple science only. I hope this was helpful. Please subscribe if it was and you want to help other people find it. You can share it with other people that are doing their science exams this year, so that they can have a useful revision resource too. And thank you for watching.